Good evening, welcome back to the STEM Shed. Today I'm going to be going over the chemistry behind the DNA extraction of a strawberry. DNA extraction of a strawberry is something you've probably done in high school, biology. But when I saw it, I saw it when I was long-term substitute teaching for a biology class. I was like, okay, so I put soap in there, I put water, I put alcohol in there. What's it actually doing? How am I getting the DNA? Well, I want to go into some of the basics of it. So for starters here, we've seen these classic drawings of DNA right here. This right here is called the tertiary structure. I call it the classic structure, Hollywood structure, but it's a double helix where it has like the little ladder looking rungs. That's what they normally show like in the Hollywood movies because it looks cool. Next up, you'll have the secondary structure. I think this is probably what maybe biochemists care about. I'm not a biochemist, but I assume this is more important than this one because this is where you can do the coding, where you'll have thymine attached to the adenine, the cytosine attached to the guanine, and remember in RNA, the thymine is actually uracil, remember that correctly, and it will bond to the adenine to become a DNA, I think. And then you have the primary structure they do, which is kind of exhibiting of the backbone, and the backbone is kind of what's important to me, what I care about. So in the backbone here, which I drew over here, and I'll go over in a little bit, is kind of thing. You'll have the phosphate group, you'll have the sugar group, and then you'll have the phosphate group at the end, and then you'll have the base here. In this case, it chose adenine, just because I liked it. And then you'll have the quaternary structure. The quaternary structure is kind of, I think, what's more important now to the biochemists and the molecular biologists, because it will show, and usually I think in textbooks you'll see like a little ribbon-looking thing, and it'll wrap like this, I think they call it histone, and it'll do this number, have some proteins attached. And if you have the coronavirus in that subject, they're using a lot of like 3D modeling to understand it better. Computer 3D modeling. So that's kind of a cool field. Alright, so what is DNA in the basic structure? So here is the backbone right here. This is the outside rung. This is like the little part they have right here. That is this part. It has a phosphate group, which is derived from phosphoric acid. You remember in your chemistry textbooks, it's H3PO4. Alright, we have and they'll usually draw this as a circle with a P in a lot of textbooks because biologists don't draw chemical structures. I don't blame them, it takes a long time to draw these. Your next one up is the ribose. Ribose is a sugar. It's also where the name comes from. Deoxyribose. I believe deoxy means the loss of these oxygen groups right here and right here. And that becomes part of these phosphate oxygen groups. And then you have your base. So, a nucleate comes from the fact that the DNA is found in the nucleus of a cell. Alright, and then you have here the base. This one I chose is adenine. And this, what you notice here is they have up here the amine. Alright, so that's amine. It's a nitrogen group with hydrogens. And this stuff right here is what makes it a base. This is a basic molecule. This, on the other hand, is acidic. So, and you also might notice here these little negative charges. These are important, alright? These negative charges give the DNA structure. The outside was essentially the outside and the overall negative charge. This base right here is what helps attach to another base through hydrogen bonding. And then when they do that, that's where you get the whole little wrappy thing. And then, of course, if you remember my polymer video, the DNA becomes actually not a forever double helix. It'll wrap around on itself and do all kinds of fun things, which makes it a little bit more interesting. Alright? So the strawberry experiment is, again, something where you'll take a strawberry, you put it in a Ziploc bag, you'll mash it up with soap and water, and salt, sodium chloride. You'll mash it up, you'll mash it, mush it, you'll filter it through a strainer. And when you filter it through the strainer, you're going to then put it into a small glass. You're going to put a little bit of isopropanol in there, or alcohol, because there's no more rubbing alcohol left. You have to actually get drinking alcohol, which they make in very high proof contents, 95% alcohol, which I had to get for this. And you do that, and it'll get the DNA congregate together. Alright, so how does it do that? Alright, well, the main thing is the soap. What is soap, and why dish soap? And what is dish soap made of? Well, dish soap, for example, this one here, is made of a lot of things. The main ingredient that does most of the work, though, is the sodium lauryl ether sulfate, or sodium laureth sulfate. But instead of drawing it out, I have it drawn right here. So I have it drawn right here. This is what it looks like. And you notice it has a sodium ion on top, sulfate right here, and a long chain. You got your ether group. Remember, ROR is ether. And then the lauryl is actually this part, which is actually dodecanol, or it comes from dodecanol. It's a dodecane in this instance. And that's where your power comes from for soap. Alright, and why soap? Well, you need the soap to break apart the lipid cells of the strawberry, and that is housing the DNA for the strawberry. You have to break it apart. So a little side note, when you're using dish detergent, detergent comes from J, or D, away from, that's Latin, and terge, which is to wipe. So to wipe away from, detergent, I guess. French might have a funny way of saying that. Alright, so why the salt? Alright, well again, I spoke earlier about how DNA has an overall negative charge. Alright, well salt is a cation and a plus, and it has an overall positive charge. So when you do this, it's sitting in there, and everything, and at this point, everything's really, really happy together. It's actually very happy, and so nothing's really messing up. So that's why you can't see the DNA yet. However, you can get this erased. As always, to get this DNA together and to get it out of that happy little soap form, you need alcohol. And here, the alcohol comes in and messes up everything, as it always does. And I can promise you, I've learned that from experience, and it is not fun. So, what is alcohol? Going back to my little blue board right here. Alcohol here, it has a ROH group. Some argue water is one. I don't think it is. So here you got methanol, has a meth group. You have ethanol, ethane group. Propanol and isopropanol. Isopropanol is what rubbing alcohol is, and it's completely sold out now because of the COVID thing. I put this here, the lauryl alcohol, just to give you a reminder that it comes from the dodecanol, which is the entail of this sodium lauryl ether sulfate. And that's just there as a reminder of that. Okay, so what happens? Alright, so if you have a sodium ion, let me go this way. A sodium ion, like this, and a plus. Alright, well water is negative, partially negative. Here's my water molecule. And it kind of sits here like this around it. And they're all around it. So there'll be, I'm going to draw the Mickey Mouse here, but you got here, H, 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 H. Alright, so you got all there, and it's surrounding that sodium. That sodium's pretty happy, it doesn't really need anything. And then again, you got your DNA over here. I'm just going to kind of draw it sloppy. Remember, it has overall kind of a negative-like charge. So, it's overall kind of negative. And this problem is this water right here is blocking it. Because remember, these electron clouds are attracted here. These are positive, but they're not. But they're holding the DNA in place. All right. However, you bring in alcohol, for example. So you bring in alcohol. I'm going to do it right here. So you bring in the alcohol, and let me go with the green here just to exemplify it. So you got the alcohol here with that long tail. Well, these are non-polar. It's just got 2 hydrogen groups here, 3 hydrogens right here. All right. All right, and then let's say you have another polar tail right, non-polar tail here. All right. Well, guess what can happen? This can work its way through here. All right. So what's happening is these DNA. I'm going to draw this down here. Is DNA, and is also happy with. I'm not going to put the lines in this one. That's kind of negative, and this is kind of positive. They are very happy with the sodium, and they come together closer. In doing so, is why you can extract. Now, in another video, I'm going to speak about the COVID thing right here, and why it's important to wash your hands, and be going back to the soap. So definitely tune in for that video later. Thank you.